will probably proceed to the next talk, which will be given by Meister. So the next talk will be about interactive visual analytics and Modelitics. And Meister, whenever you will be ready, the stage is yours.
I start, by the way. Okay. Thank you very much for inviting me to this summer school. It's a fresh one. I mean, it's at the end of the summer. This is a summer school on BioMedVis, and I will sort of be on the Vis side. And the title is Interactive Visual Analytics and Modelitics. Maybe you have heard about visual analytics. It's an interplay between an automatic analysis of data, information, with an interaction with the data, so you would like to have the human expert, the human in the loop. And secondly, visual analytics is about analyzing the data and finding out some more information about it. But we are right now also in a stage where we would like to synthesize elements like larger biological structures, cellular structures in this way. And this brings in this Modelitics where you combine automatic algorithms, techniques, and the human in the loop and some interaction to actually get some objects out of it, cellular structures, for example. So I have here 3 examples where this visual analytics plays a role in decision-making systems where you have alternatives, flooding scenarios that you are investigating. I will not talk about that. The second one is about cohort studies. Lars will talk about that, and maybe Renata, I will also not talk about that. But I will talk about these cellular structures in structure and integrative biology, and where a colleague of ours has been doing his PhD thesis. So let's get started. Maybe I have the wrong one. I have 2. So maybe this is my last slide. Oh, you manage? Maybe I'm too weak to press the button. Oh. All right. Thank you very much, Katka. She showed me the right button. So visualization. I mean, visualization is about computer-supported interaction with visual representations of abstract data to amplify cognition. So this is a traditional definition, and it's about data, and you would like to know something more about the data. But it's not just for fun. You have a specific task, a specific goal that you would like to solve. So you would like to be brighter, more intelligent afterwards, and you're using these visual representations. And data is the central concept, and therefore the various branches of visualizations have been named after the type of data people were investigating. It could be volumetric data, 3D scalar field, for example, flow data, vector data, or abstract data, information visualization data, visual analytics data, or the area, or visual data science recently. But the interesting thing is the data is increasing in complexity for various reasons. I will explain that in a moment. And as the data is increasing in this complexity, you have to do something about it. I mean, you could make the visual representations more complex, but your human expert is not getting much more intelligent in the last couple of thousand years, for example. So you cannot cope complexity, the increased data complexity with an increased visual complexity of your representations. You have to do something about that. First, I would like to explain some concepts: visualization, visual computing, scientific computing. And usually there's the visualization pipeline. Who of you is from computer graphics visualization? Most of them. Oh, so I'm in the right place at least. So the visualization pipeline is a sequence of steps that people are doing from the data acquisition. Then you do some post-processing, you remove errors, you do some filtering, and then here's an interesting step. you map your data to something that you can perceive. That can be color, that can be transparency. And afterwards, you do a rendering, you make an image out of it. So that would be the typical computer graphic techniques that come into play. But sometimes we are doing all these steps, but we are rather interested in a quantitative analysis. If you do imaging, as was Katka explained, then the doctor would like to find out how large is the pathology. If you have a blood vessel and you have a stenosis, how large is the stenosis? I don't want to have an image of the stenosis, but give me a quantitative number, and because of that, I can think about different therapy steps. And so this entire pipeline is embedded into scientific computing and visual computing. So how is science done? I mean, you can do a theoretical science by formulating theorems and proving those, or you can do experiments, or you increasingly, that's interestingly done in computer science, you take a simulation and you do the simulation instead of an experiment. And whereas in the last century, physics was the driving discipline in many areas of research, nowadays it's rather computer science, because you can now simulate phenomena, and that's really great. But on the other hand, you do the simulation here, and after the mapping, you somehow have geometry, you have visual information, you have to handle those. And that's visual computing. And visualization, as we see it, is part of this visual computing, where visual computing deals with acquisition, representing the data, processing, analyzing, and increasingly synthesizing the data. Nowadays, people are talking about digital twins, so that you would like to have, you have a natural phenomena, but at the same time, you would like to have a digital representation. You have to model that, you have to define that. And so increasingly, we would like to have a synthesis going on and usage of this visual information. And visual computing is a lot. I mean, so that's an interesting discipline. In Vienna, we have an entire study curriculum on visual computing, and includes computer graphics, computer vision, visualization, and all the way. You can have different sensors. Interestingly, you have virtual and augmented reality. Nowadays, people are talking about immersive analytics, situated analytics, and basically visualization is also a HCI, a human-computer interaction discipline. Okay, so how is visual computing embedded in science? So what scientific computing is doing, instead of doing this costly wet lab experiment, maybe you have a simulation and you have a virtual petri dish. But there was Richard Hemming. Who of you knows Richard Hemming? Personally, I suppose. I mean, although he passed away a couple of decades ago. So he's this guy with this Hemming distance between codes, the distance between codes. And he was also saying the purpose of computing is insight, not numbers. Computers are really great in doing calculations, but then you have to make sense of that. And then you need the human expert. And you would like to get to know more about the data and not the numbers by themselves. And now when we are talking about visual computing, where you get images out of it, the images by themselves are not the end goal that you have. And in this case, also the purpose of visual computing are not images. You get a lot of images out of it, but again, you need the insight, the analysis, so that you can make decisions that should be actionable in a way, what you get out of it. And so if you do a real experiment in a petri dish, you have a microscope, you have to take a look at it. Or if you have a virtual petri dish, you need visualization, visual computing, and that you make also a visual representation out of it. And this is then the computational sciences, where you replace the natural phenomenon with a model, with a digital model. And nowadays you can take any disciplines basically in research and you put computational in front of it, and that's something that that's happening. It's computational mathematics, computational economics, computational law, computational journalism. Like you can do many things. Computational archaeology. You don't have to excavate the object. There are scanning technology that tell you what's under the ground, and it's kept there. and you get a 3D model, and you can reconstruct which structures have been there around. So then there are these terms: big data, smart data, visual data science. So the data science is sort of the next type of how to do research. With computational sciences, you need an expert that is coming up with a complex model, a weather model, for example, climate model, and then you're simulating that. Data science is based off, you have so much data, maybe you are dumb, but you know how to quickly access the data. And by knowing much data, you can also have a model that helps you. Like for weather prediction, if I don't have any idea about precipitation, pressure, whatever, if I have collected weather data over decades, I'm just looking in the past, where has been a similar situation like today, and based on that I can make a prediction. So the more data you have, the better. There you have to have mechanism to quickly access the data and index that, so there are different challenges there. And data is increasing in complexity. Well, the volume is increasing. Well, that's has been around for decades, but also the velocity. We are now looking at real time, at dynamic data. Katka was mentioning there are medical imaging systems that can measure dynamic objects. I mean, you can have a scan of a beating heart, for example, that is not providing one volumetric data, but hundreds. Data veracity, how certain is the data, how uncertain. In the past, this was a little bit simpler. If you had a lab experiment, all the conditions were fixed, you could control them, and you knew the accuracy, for example. Nowadays, you have so many different data sources that are in databases. You don't really know who took them, what's the accuracy, how can you trust this data. So data veracity is a big challenge. Data has a scope in time. I mean, it might be valid for a certain moment in time, but less reliable at a different time and space. And the data variety is also a very important aspect from homogeneous to heterogeneous data. Nowadays, you have, for the same phenomena, very many different data sources. How are you combining those? And what are you doing if they are contradicting each other? Or Katka was and Jan were pointing out that you have so many different scales on a phenomenon. But when you switch from one scale to the next, when is one scale valid and not valid anymore? And when does the other scale kick in? And if if you are in between two scales, which data to take? So this is very interesting. So the data, if you look at at one item, it can be simple or complex. Or how many items can you have? You can have a single or many items. And if you have a single and simple item, well, this is a number. This is a special number for me. I mean, I'm not telling you why. But, you know, if you have just a number, why you don't do visualization. I mean, it's why bother with it? But if you have a simple data, but many of those, well, maybe you do some information visualization, scatter plots. You have attributes, and on the axis, you show two attributes in this case. You get a point set. Maybe you get clusters, you find outliers. So this is interesting. If you have a complex data, but a single one, that was something like in the past you had in the medical domain, you were scanning one specific organ and you were thinking about complex algorithm, and then you ended up with what was considered scientific visualization. Nowadays, increasingly, you have complex data, but you have many of those. So if you don't have one volume, you're doing a cohort study. You are investigating a patient and you're taking scans, 10 scans, 15 scans, or you're taking an entire population and you're population regularly. How can you show the differences in this data or the trends, for example? And so this is an interesting part where the data heterogeneity comes into play, and you have spatial and abstract data that you have to visualize. Let me give you an example here. This is an example in one of the books of Eduard. Who knows Tufte? Yeah, maybe even personally, he's still alive. And what he's doing, he's showing here is the interplay between abstraction and showing the context. Here, he found an example. You show the length of rivers, and you are lining up these rivers. You take a river, and you sort of straighten the river, but not fully. So you keep the small bends in the river and maybe some lakes and annotation you keep there, but the large bends you're taking away. So in a way, it's this is a you're throwing away in the spectrum of the bands from a sharp band to a large band. You are throwing away the large bands, for example. And this looks like a bar chart in a way, and you see the length, but you also see many other information. And here you see a close-up. And this is something that you always have, in many cases in visualization, have to think about. How much abstraction can your spatial data sustain, and how much space does your abstract data need? So in that clearly depends on the task, on the goal that you want to achieve. Here, the length is a primary attribute that you would like to convey, and therefore this lineup makes sense. But you always have to think about when you are throwing away too much context, you might lose information as well. So I'm showing an example that is in line of this straightening, and that's about polyp detection. I mean, here you see, you can do a colonoscopy. Who already did a colonoscopy? I did one. I mean, maybe I'll show you afterwards some images. I think a virtual endoscopy would be much more pleasurable. It's not a pleasure, but I think many of you will enjoy that late in your life. Beyond 50, you'll get a recommendation to do it. So this is you are scanning the column, and then you're moving with a camera through the column. But actually, you're restricted because you cannot straighten the column of the patient. Patients don't like that. But virtually you can, and here you have one image that shows you the entire situation. When you're moving with the camera through the real column, you have a limited view frustum, and you actually have to move in both directions to see everything. But so here in this case, you unfold it, and then you actually would like to see these polyps, and the size of the polyps tells you if they might make trouble or not. So this is an example of this, how much abstraction do you include in your visualization. And this is an interesting area where there are several techniques in this area, like visual analytics or Modelitics. Comparative visualization, you will hear several talks about that. Quantitative visualization. Quantitative visualization means you see a number or a quantity in your visualization, not something. In most cases, visualization is about qualitative information. You see a pattern, but you cannot fully delineate where some object starts and the other stops, for example. And with quantitative visualization, this is a step in the direction where you would like to be able to then also give direct numbers. Scalable visualization, I think I will talk a little bit about it. Now that the data is available on so many different scales, it's interesting how you can integrate those scales. When I'm interacting with an organ, how does this interaction translate down to other scales? Is it at all possible to do this? Something like that. And linked and integrated views means that if you have so much data, you can make several views of the data, and on the one view you show specific effects on the other attributes. Or integrated views would mean that you are including in one representation additional attributes. In immersive or situated analytics, you could have this room, and projected on the walls is some information that you usually don't see, maybe where the wires are behind the wall. So that would be an integrated view where the several data sources are combined. In data and computational science, sometimes you have a model, like a climate model, and then you have data as well, like an airplane is going and the airplane is measuring the temperature at a specific spot. Does this measurement correspond to your computational model? You have to do what is called data assimilation. You are combining the data that you are measuring and your simulation that you have. All right, so these were general remarks, too long as usual. And now I would like to go into a specific topic, and that's integrative cell biology, which treats the topic of generating models of larger objects like viruses or entire bacteria. And this is the work that a colleague of ours, Tobias Klein, has been doing with Ivan Viola, who has done a lot of work in this area. And there's even a spin-off company, Nanographics, that is dealing with such topics. And here you see a part of a cellular structure, and it's visually very complex. And the question would be, how can we model such structures? And from our topics that I mentioned before, it's about visual modelitics. It means that you would like to compare different of these structures. It's for sure in the scalable visualization domain. These objects contain billions of atoms, and you cannot render all those, but most of them are not visible from a certain perspective anyway. How are you handling that? And then the data and computational science means you are measuring, maybe you have an electron microscopy data. This is your data, and then you have a sort of a model, and how are you combining those to get a more refined model? So this is an interesting topic. And molecular graphics is has been always one of the guiding domains for computer graphics. I mean, the defense industry, the movie industry, advertisement, the medical domain were other areas, but molecular graphics was also very, very important. And like Watson and Crick, when they came up with their double helix model, they really built a physical model. Nowadays, again, you have the term physicalization. This is going back. You take your digital model, you print it, for example, but they started out building real 3D models and looking at those. Or this was a different model where atoms were represented by spheres, and you could see these strings. And so for building such models and looking at them helped them to reason about that. And early on they had graphic systems, and the bonds between the atoms are shown here, and they had an interface, sort of a half sphere with which to rotate the object. So this early on was very important. And then have been illustrators like David Goodsell, for example, who is drawing fantastic 2D images of cell and cellular structures. And his in watercolor, for example, takes him very long time. And some of these strategies can be taken to the computer and automate. And what's an advantage is also that we can go also to the 3D thing, whereas David is usually making 2D images. The third dimension can be used as well. And there are systems around. I've just mentioned a few of those that take information and make molecular representations out of it, like the structure, secondary, tertiary structure that Jan was talking about. This is about the setup of an individual cell, and here this has been a work in our environment to rapidly, fast render objects that contain billions of atoms, for example. And I was talking about big data, and why is it in the molecular domain an interesting area? Because the data is exploding. I mean, this is just a a protein, the PDB, a very well-known protein data bank, and you see how sort of exponentially the data that you get into it is growing. And then you would like to make a model, but the knowledge is changing very fast. Maybe you have suddenly new proteins, new information, and you and then you would need to do your model from scratch again. And so this can be very cumbersome. And here's the scale of how you can look at the data and what different possibilities you have. These are cells. Eukaryotic means they have a nucleus in it, all down the way to individual atoms, and there are scanning mechanism for various scales. You can look at the objects, but I mean they have to be rather large. Then you have the light microscopy, then you have the electron microscopy, cryo-electron microscopy has been mentioned by Jan. You have to freeze the object. You're losing a lot of information. The whole dynamics is gone. Then you can have X-ray and nuclear magnetic radiation information, so that you get information about this. But there's somehow in between, there's a gap. On the one hand, you can take a look at the larger structures. You can take a look at the small structure, but only at individual elements, but not to assemble them. So we have here sort of the nanoscale up to the microscale. And on the nanoscale, you're looking at an individual molecular, maybe at the macromolecule, and that can be done maybe also through simulations. On the larger structure, you look then with a microscope, but it is this in between where you have two situations. Okay, so nanostructure is fine, and here we see a video where they're playing around with individual atoms. So they're moving around the atoms and assembling them in a way how they would like to have it. Also on the larger scale, you have the internal structures in an individual cell, a cytoskeleton, for example, and that gives you the information what's going on in the cell. But here in between, you have the problem. You have either sparse models. A sparse model means you have only an individual protein, or you have fuzzy observables. Fuzzy observable means that here the cell component is so small that your scanning algorithm is just giving you a very rough, fuzzy information, but you would like to combine those. You know, maybe from here, from the fuzzy observables, you know how much objects of a certain kind are given, but you don't know where they are. On the other hand, these sparse models give you an information how they look like, but you would like to place those. That's where these computational models come into play. And the general strategy is compute, not store. I mean, you could make a model and store the entire model. Then a new knowledge is coming up, and then you would need to change your model again. So the idea would be sort of to have a recipe, or in computer graphics, this is called procedural modeling, that gives you an algorithm, a way what you need to do to come up with this model. And if some of these recipes change because a new knowledge is coming up, you just need to change the recipe and you can do the recipe again. So this is a scan of a virus, a very fuzzy one. You could take with computer graphics, you could do some coloring, or you could populate this space with individual elements that you know are given here. You have the membrane, for example. If you look closely here, you see the individual parts, and suddenly the color is changing. So if you're going from one scale to the next, it might make sense that you change the coloring accordingly. Here we see individual atoms that describe what type of atom is given here. If you're looking at it from a larger perspective, it might be interesting that entire compartments of the protein are colored. So this would be an example of a scalable visualization. Depending on the scale, you need to change the coloring. So this is a combination here on what I said before. Maybe you have an experimental technique, you are measuring a structure and you get a rough outline of it. These are my fuzzy observable, it's very fuzzy. On the other side, I have a very detailed information, and then I like to have a recipe that tells me how much of this individual information might be given here. And so this is a modeling approach, and people at the Scripps Institute in the US have done this. On the other hand, it would be nice to have immediately an instant model that you can take a look at it and that you can play around with it, that you can change the parameters and immediately see how this object might look like. And that's on the one hand the information that you get from the structural biology, and on the other hand scientific visualization tells you how you might render those. And what Toby was doing in his PhD was sort of the instant construction and visualization, make this really also the modeling possible very fast. And that's the Modelitics part. The recipe that you get from structural biology gives you information. How do you need to put these things together? Well, give the current knowledge. And then you can drive the modeling process with it in the sense of how you're populating that, how are you populating the membranes, for example, and to do this in a fast way. So he has basically, there were 4 steps in his work, and I will in detail only look at the one. It's the 1D. You have the DNA strands, for example, that Jan mentioned. These are very long, thin strands, and how can you generate those very fast? You know, in a way, you use a random walk process, but if you do it sequentially, it can take very long. The 2D one would sort of be to generate the membrane that is including a bacterium or also a capsid, for example. Then you have soluble elements inside. Where are you putting them? And the 4th one would be to include also dynamics, how they're moving. And here you see a DNA strand generated sequentially, can take very long. This would be the membrane, a bilayer of lipids that are encapsulating the cell, for example, or part of it. And then the 3rd step is how am I placing the interior elements? And then the last one would be sort of the dynamics here. And this is a very dense environment, but it is most of the elements that are here in the cell are not showing at all. I mean, there are no hydrogen water molecules. You leave away all the water molecules and it still looks very dense. And if you are placing the parts here, you have to make sure that there's no collision going on and things like that. And so the general strategy is compute instead of store, because if this the recipe corresponds to the current knowledge and is changing, you can update the recipe and then automatically through Modelitics, the new updated model is generated. Sometimes there's a missing knowledge. How are you representing missing knowledge in your model? So this is an interesting topic. So let's take a look how we can construct these very long DNA strands, for example. So the DNA is really very, very thin. I mean, you can't see it, but if you take a DNA strand, it has a length of 2 meters. And or if you take the DNA strand of a human, it's a 71 Earth-Sun round trips. I mean, until tomorrow you can check if this really is the case. I mean, unfortunately, I did not check. I was trusting Toby, and Toby was trusting this web page, but maybe this is not true. It's just 70 times. So, but please check it. So it's a very long strand, and here it is shown how it is generated. It's basically a random walk, and it is avoiding other elements that are in the in this cell, so to speak. So the general strategy would be you take this cell, you populate it with elements that are so dissolved in it, and then you have this linear strand, and you do a random walk and avoid other elements. But this is sequential and takes very long. And the principle is, how is this random walk done? You can measure how wiggly the DNA is. So you have a flexible DNA, which means it's making a lot of turns, and you describe this by a distribution. So if you would think this is the top or the one end of your DNA strand, then you can think of a half sphere, and on this half sphere you have stochastic information that describes in which direction you should go. And if you are very flexible, you can go in any of these directions. If you have a stiff DNA, which means that if I'm going in this direction, I'm not changing the direction very much. This is called the persistence or persistence length. You have a much smaller distribution, spatial distribution, and then sequentially you are calculating that, and if you have a collision, you are avoiding that, and so you generate that. But if you have millions or billions of these steps, that takes very long. And you do this random walk, and then you're populating the entire strand with your amino acids. But how can it be done as well? I mean, we are looking at a parallel approach. I mean, there are some restricting assumption, and with every model, you can only to a certain extent regenerate what the original phenomena is doing. But we are doing here something that has been done in fractal geometry to generate landscapes, for example. Do you know fractals? Personally, I guess. Yeah, you have fractals inside yourself. Your lung is a fractal in a way. You yourself, you don't have a dimension of 3, but just 2.85, something like that. But what we are doing here is sort of midpoint displacement. We would like to have a strand going from here to here. We take the midpoint, and we are displacing this midpoint, but this displacement has to correspond to many of these individual steps. And if you do 1,000 of these steps at once, the variance is much larger. So for the midpoint displacement, you take a variance in your stochastic process that corresponds to the length. And the next step would be, you are doing again a midpoint displacement, and here the parallelization comes into play, because here suddenly if you fix this position, you can generate the first half of the strand in parallel with the second half, and that's actually the approach that has been done, and then that leads then to the speed up by generating a linear structure in parallel. What might happen to you is a collision, but in computer graphics, there are physical engines included, and that do some collision detection and collision handling, and that's also what is done here. Basically, you overlay the entire structure with a grid, and then you can do a collision detection. You find out, is a collision happening? You resolve this collision. This might lead to a collision somewhere else, but then you can resolve it there as well. So we see it here in how to do it locally, and even if you have a larger structure. This is the DNA of a bacterium, a very large one, and in red dots you could see where collision happens. And if you do an instant calculation, you can change the outer shape immediately, and instantly you get the result. So here we are playing around with several parameters like the persistence or how long this shall be. And if you change the initial parameters, you immediately get another specimen. So sometimes you are interested in having several of those. And here the step length, or how many steps you have, is increased. And you see when it was necessary before to spend seconds on it, in part of 1 second, this large strand can be generated. And so I think here it was playing around with the persistence length that describes how stiff and how wiggly the result is. Okay, here are several of these structures with that have been generated. Escherichia coli is a bacterium very well known. The Sorangium cellosum we have seen before. This has the largest known genome in a bacterium, for example. This one can be generated, well, almost a second, but before it took minutes to sequentially generate this structure. So there's a nice speed up, but there are some limitations. You can read up the paper which is given in the reference. How are you evaluating that what you are generating corresponds to reality? Jan was before referring to also to a neighborhood calculation. So in this case, if you have a structure and the wiggliness means that that parts of the object that are far apart, if you go along the linear path, are then suddenly close together. And this is mapped into this distance in array. So if the red and the blue part are coming together, here we have linearized the entire structure here as well. And if they are close together, I can color here this spot, and then from these patterns and how dense the areas are of the diagonal, I can sort of statistically evaluate if the wiggliness of what I have generated corresponds to what I have measured. So this would be the data science part. Okay, maybe I'm showing here a short video. This would be with the sound.
We present a novel approach to integrative structural modeling of the biological mesoscale. These complex models can comprise up to millions of molecules with defined atomic structures, locations, and interactions. However, the creation of these types of models has previously been a laborious
the 4
process
steps I was
and a
just
bottleneck in study.
before pointing out. We see here a virus, and there are 4 steps. The first thing is, well, there's the membrane, there are the solubles, and they consist of various compartments, such a structure, this membrane has to be generated. Here, sort of a texture synthesis approach is used. Then you have the solubles inside. Here you have sort of a volume filling approach. Then inside you have the linear fiber of the DNA that has been generated. There's a technique in texturing that is called Wang tiling, and you're taking a small piece of the surface and you're tiling the entire 2D surface to get an irregular, non-repetitive pattern on it. For the soluble components, you are taking a grid. I think this is 3 steps that you're doing. You have this grid. In this grid, you put your individual objects. Then you have the collisions that you have to resolve. And with changing the parameters, you can fastly generate very different objects. And the step 3 would be this collision detection and avoidance. Here in red, you see the individual collisions in parallel. Again, you always like to do this in parallel. And there are some, sometimes you hierarchically, you take some objects together to do this more efficiently. So these are these proxy geometry. And here you see the fiber structure, but in this case it was sequentially generated, and that took the longest time, and therefore we have come up with the parallel version. And here you see blood plasma. This is a test of how many objects you can generate and render. This is an HIV virus that where you have generated all the individual elements, the inner elements, the membrane, and the fibrous structure. This is another one. which we have seen before. And yeah, that's what I know about this. Okay, so I think this was a quick explanation of this area, an example in this where you have a lot of spatial and abstract data, and you have these interesting areas in visualization where you can work at. So at the end, I can give you an outlook of interesting topics. Maybe I'm not going through all of those, but it's interesting in comparative visualization. How do you define the metric? What's the difference between an object? What to compare and how to compare? And this clearly depends on the task and goal that you have. I mean, if you would like to compare the length of a river, it's good that you maybe straighten it, but there might be other measures of how to compare rivers, many more. Usually, we very often have a multi-objective visualization. You have several competing goals. Here we see a simulation of a lighting in a workplace. You would like to have as few light sources as possible. You would like to save energy, but on the other hand, there are some regulation by law how bright the workplace has to be. Where am I positioning those? And here we see again various alternatives that are possible, and you would like to quickly look at those. Multi-scale visualization, a very interesting part. I showed you some examples before, and also uncertainty has been mentioned, and that is handled in this recipe approach. Maybe the recipe is not complete or totally correct. Do you have a simple mechanism to change the recipe, and the system will automatically update the model? What is happening if you have contradictory visualization? Your data is clearly contradicting each other. Maybe because for one data, the scale doesn't work anymore. What are you going to do about that? Quantitative visualization and interaction. Can you repeat that? I mean, can I select 10% of my data? So that would be a quantitative interaction. And very interesting, the combination of data science and computational science. You measure the persistence in your object and you use that in your recipe. We talked about that also. If the data is too complex, you cannot show all the items. You could do a statistical visualization. You just show the median, the average. But then there's something in between, and that's the ensemble visualization, which we will see. Sometimes you have parameters and you're not sure which values this parameter have. So parameter-based visualization is interesting. Computational steering and visual steering means you look at the result immediately, and therefore the visualization has to be fast. You change a parameter and immediately you get an update. You're changing the size of the membrane and you would like to see the result immediately. Visualization in the wild, very interesting nowadays. You don't have a data that you measure in the laboratory. You have 20 different data sources on the internet where you're not sure about the quality. How can I combine those? What's the scope of the individual data? Which data set to trust more? There are terms like data fabrics in data science. You are mixing together various data sources. Data-driven visualization to task-driven visualization. For certain problems, I don't care where the data is coming from. I would like to know what's the weather tomorrow. This is my task. And the system shall find the best available data and combine the sources. I mentioned this visual modelitics, going from the analysis process where you are picking the object apart and looking at the pieces. How can I put it together to make a synthesis and supported by the computer? I know that my cell has a lot of individual elements. I don't want to control individually where to place them. I give just some constraints and the system shall do it. Well, machine learning and visualization, we will hear talks about that. And immersive analytics is and situated analytics combining virtual and augmented reality with visualization and physicalization. I think Renata will afterwards talk about that. not having something on the screen, but really tangible that you printed out, for example. At the end, do I have time? Then I can show you another video where the music would be great. Here I will sing. I mean, it's and here it's also about visualization to illustrate something. And here the point is nanographics in beta-Mindeq have made a very impressive, at least to our understanding, animation of the COVID virus. And this video is illustrating the way from sketching it to coming up with a result. It's not a spoken sound, but music, which makes a lot of a difference, but we will do without. And so you start out with sketches, for example, 2D rough illustrations and maybe coloring a little bit. And from that, combining it with some measurements that you have been doing. So he was taking cryo sections that were generated by a group in China. And maybe we see those. Oh, here we see those. So this is combining data science and computational sciences, taking these cryo sections and then making a model and then coloring it. Although, you know, you would not see the color here because it's too small. And again, taking some information that is available. There were also new knowledge describing how these spikes, how mobile they can be. And so this is information that changed over time. And with a recipe, you can easily include that. If you would have generated a model, then it's outdated all the time. David Gutzel was saying, whenever I'm painting a new illustration, it's outdated because things have changed so much. So this brings me to the end. And I guess in this area, what's really interesting, you always, like in computer science in general, you have to think about abstraction in the simplest way. What can I leave away? So to emphasize what I would like to convey. But you cannot abstract too much because that would leave away the context then. And so there are a lot of people in the acknowledgement also from the group here that were actively contributing or driving this research. Thank you very much.
Thank you very much, Eddie, for the great talk and very impressive results you were showing over there. So let's go to the questions. Are there any here? And meanwhile, just, yeah, please. Just a second, I will just give you the mic.
Thank you. So thank you for the very great talk. I have a general question on the interactivity of the visual analytics. So it's like when I am developing a visual analytics tool, I always add this function, add that function, and I keep adding, then it becomes a bit too much. I'm wondering, would you have any tips on the balance of offering a lot of possibilities or the core idea, to keep the core idea?
This is a very good question, how to cope with this and not making it too complex. For visual analytics systems, sometimes they're talking about dashboards. You have a very complex data set, but in a concrete application, your dashboard is very simple. It's reduced to the few things that you have maybe in a power plant. In a power plant, they also have physical dashboards, and they have only a few of those. So that's very interesting to think about how much complexity shall I add. And you also think of in economics, they're talking about Grenznutzen, the limit gain that you get of it. So if you're adding some more information, how much gain do I get? So I think here it would be necessary to think about that. What is good enough? So, but this is very difficult to answer, and that might also be afterwards. You could do some studies, for example, which part of your interface are the people using regularly, very often, very rarely, and then there are also some mechanisms to simplify the interface, for example. And in the 3D spatial domain, there are generic techniques like hierarchical techniques, occlusion techniques that try to simplify what you're showing to the person. I mean, if you're showing a too complex interface, you have to think of the perceptual and cognitive load that you're putting on a person.
Thank you. Thanks a lot for the question. Are there any questions here in the audience? Okay. Thank you.
My question would be, how are these visualizations used in practice, for example, in medicine, when we are combining different modalities and, as mentioned, also the uncertainty is increasing? For example, the virtual endoscopy, is it really used or is it accurate enough?
I mean, it makes a difference. Clear. Some of these systems are speculative, even if you include all the current information, and you have to be really careful if you are applying it to patients, for example. So in the endoscopy domain, I think there have been studies in the US, in the military, where they did screening with virtual endoscopy. The big, well, the most unpleasant thing with colonoscopy is the cleansing. So you're drinking some stuff and then you go to the toilet and then your colon is empty. And with the virtual colonoscopy, you cannot avoid this step. Also there you need a cleansing of the colon. So sort of you have this unpleasant thing. And the second disadvantage is if you find a polyp, then you cannot immediately remove it in a virtual colonoscopy. So in that case, you would need to go in. But for the colonoscopy, they have been doing, I don't know the exact numbers, but thousands of people that have been investigated with that. Generally, in the medical visualization, it's clear there have to be done very elaborate validation and verification studies. And that takes very long because if you go from an experimental stage to a clinical routine, there are in the US the FDA agency that has to approve these procedures. I think we started 20 years ago with blood vessel curved planar formation, cutting the blood vessels, and it takes very long time. And I mean, it's used in a clinical setting where the patients have to sign, you are now treated with something that is not approved, but that takes very, very long time. Yeah.
Okay. Happy with that? Okay. Any other question? Because if not, Meister, your colonoscopy also raised some interest at Discord, obviously. So you promised that maybe you will show us some pictures afterwards. So we are looking forward at some point.
Okay. I don't know if this is indecent. When I had my endoscopy a couple of years ago, I really had it on my desktop. I don't know if it's appropriate to show your private parts to.
Maybe we can keep it offline afterwards.
Okay, I will. Whoever's interested, approach me. I will show you images from my colonoscopy.
Thank you, Meister. We really appreciate your openness. And thank you very much once again with this small thing. I know that you are not the biggest fan of chocolate, but you can share it with us. Thank you. Thank you once again.